And then you all retook that quiz online, right? Okay. We did it on paper. And then
Did we have to take it again?
I put it online on Blackboard and said, if to record your grade for this quiz, take it online.
Is it still up?
Yeah, it is. Just want to log into Blackboard once in a while. I'm pretty consistent. I'm trying to be really consistent about keeping all your stuff on there. Man, everything over here is ice cold. Okay, we left off right after carbon, right? We talked about carbon. Talked about stereoisomers. Don't let this chemistry stuff scare you right here, these functional groups. This box down here, FYI only. You'll see these words a bunch of times. If you don't ever stop and figure out what they mean, they'll never mean anything to you. They'll never help you. So that's really just to help you commit on the memory. So if you see it in something later, you might have an idea of how place to go back and find it. But I'm not going to test you what a ketone is, okay? We talked about dehydration and hydrolysis. It'd probably be important to set in your mind that dehydration reactions are building molecules. Dehydration reactions are building molecules by removing a water molecule. And then hydrolysis is breaking apart big molecules or polymers. Do you all have your slides that I printed out for you? I've got extra copies here too. You want to look at those while we're talking. Find a good marker in here. Okay, we covered carbohydrates. I'll start there again. Carbohydrates are a class of the four major molecules we're studying. Remember, we got carbs. In the order of the book, it goes carbs, nucleic acids, proteins. I can never spell proteins right. It's either IE or EI. Don't judge me on that. And lipids. These are the four major molecules you want to know about. We talked about carbohydrates and we talked a lot about sugars. What else do you remember about them? What do we have carbs for? We talked about what are they good for? What do you eat before a football game? You go play. You load up on what? Carbs, right? Energy source, yeah. A good number to remember is carbs have about 4 calories per gram, if I'm not mistaken. It might be. Let me look at that. It might be gram. Go back here to the lipids section. Kilocalories. It is gram. Okay. Let's just spell it out. Kilocalories. Okay. And number 4 will come into play later. You want to attach that number. So calories are the basic unit of what it takes to raise 1 cubic centimeter of water 1 degree, basically. We also measure energy in our food sources by calories. A kilocalorie is what we'd say is a big calorie. It's 1,000 smaller calories. But there's 4 kilocalories in 1 gram of carbs. You can contrast that to what we'll talk about later, which is fats and lipids. There's 9 kilocalories per gram. So it's double and a little bit more. What? No. It's out of balance. Try that out. If it don't work, I'll give up. Okay, carbohydrates in their simplest form are made up of sugars. Let's get our sugars right. We talked about them, some of them being stereoisomers, structural isomers. Okay, so when we're talking about carbs, we have to talk about sugars. And we had monosaccharides, disaccharides, and this could be a single glucose molecule, right? Mono. And a disaccharide could be two or more, or just two different sugars, such as glucose and fructose. Take a second, look in your slides at glucose and fructose here. I'll find the picture for you. Page 5 in your slides. And you're looking at slide basically 17, 18, 19. And you can see fructose and glucose right here are structural isomers, just to kind of drive that idea home. And then galactose is another sugar that can be linked up to glucose. But all three of them are related in their carbon skeleton. Just the arrangement of some of those hydroxyls or the hydrogen ions make them different. But when two of these link up together, it's a disaccharide. And so human beings, we usually have glucose as our main source to make ATP. And it can float around in our blood, no problem. But in plants, their sugar is fructose. And they transport their sugar in a disaccharide form. You remember how I said some sugars are transported as disaccharides last time we met. Together, just FYI, it says, this is straight from your book too, these two sugars are called sucrose. And this is the same as table sugar. Okay, so plants need to move their glucose around with the helper, fructose. But human beings, and animals, we can move ours around as just glucose itself, or chains of glucose or whatever. Okay? So we got these basic building blocks. We can put a bunch of them together and make these long polysaccharides, many sugars, right? I'm not going to write that one up there. Okay, and we talked about the forms of polysaccharides. So we get a bunch of these linked up. We got long chains of sugars. All right, I'll go ahead and write it up here. Polysaccharides. What were the hallmark examples of those? They're right there in slide 20. Starch is one. Right? These are plant forms, and these are animal forms. Okay? Long chains. All right? The way these polysaccharides link together, these disaccharide chains connecting to these, will determine how these sugars are linked together. And they can be linked together with these things called alpha or beta linkages. I don't want to get in the weeds on that, but depending on which one it goes, will determine glycogen versus starch. Okay? Remember, we can't break down this starch, these carbs, because we don't have the enzymes to break them and get the energy out of them. But cows do. The bacteria in their stomach, maybe some protists, I think, are also able to break down cellulose, release energy the cow can use to do cell work. So we have to eat the cow to get that energy. A third one is called, also, let me call out that cellulose is more for support. Okay, it makes up the cell wall in plants. Starch is more like what it can break down as energy. Okay, So carbohydrates have two roles, energy and structure, all right? Energy storage and structure. And then the last one, the last form of this long polysaccharide chain is called chitin. All right, and that's like the hard exoskeleton of insects and lobsters and things like that. It's shiny. Shiny and hard and structural support. It actually has a nitrogen containing derivative of glucose in it. Okay. I didn't mention amylose, but that's like a precursor to these. And amylose pectin is another precursor on top of that one, like these side chains start forming. That pretty much covers it for carbs. They're simple sugar components, make long polysaccharides for energy storage and cell structure, plant structure. And since they're all basic building blocks, it's how they're linked together in their chain form that makes them do different jobs. Any questions about carbs? Okay. I'm going to erase this thing. I should have cleaned my board. What's the next one? Yeah. What makes up a nucleic acid? A sugar ring, a phosphate. That might be the correct chemistry term, but it's a phosphate or a phosphorus group. And the backbone. Let's look here. On page 43 and 44, it says, DNA and RNA are made up of 3 elements, a 5-carbon sugar, organic nitrogenous base, and a phosphate group. Okay, this is a 5-carbon. How many carbons are in the glucose we've been studying? Remember the formula? C something.
6.
Yeah, 6 carbon rings. Now we're talking about a 5-carbon ring sugar. Okay, and there's 2 flavors for DNA and RNA. There's ribose and deoxyribose. You'll find ribose in RNA and deoxyribose in DNA. Okay, when you get all these together, you get what we call a nucleotide, and in genetics, that is the GCTA, guanine, cytosine, tyrosine, adenosine, and uracil, which is found only in RNA. Did we do the protein building exercise, that worksheet where you had to like transcode all the amino acids and stuff? We might stop and do that together as a project. Okay. 2 of these have like a big base on them. and we classify them as something called purines. Okay. Adenosine and guanosine are what we call purines. And I'm going to have to look up how to spell pyrimidines. Let's see. Okay, that's important to remember for your upcoming exam for this unit. You just have to commit that to memory. I should put uracil on here. Okay. If you look at your picture on the next slide, you can see in slide 24, that nucleotide structure, which is adenosine, I think, has a double nitrogen ring base. Some of them have double, specifically the purines. These over here have a single ring in them, if you were to look at their structural formula, and that's on page 45 in your book. What's important to remember is that when these things come together to form our DNA, the long polymer, all right, the way they link up is important because DNA has to have a strand that is read this way and a strand that is read this way, right? Well, how does that happen? How do you get two different ways going in different directions? The polynucleotide chains have polarity, meaning the two ends are different. One end has a phosphate and the other end has an OH group from the sugar. And we call those the 5' and the 3' ends. Okay? So 5', which is sometimes written as 5 and a little hash mark at the top. That's the phosphate. And 3' is the OH end. And because it has these two ends, we can flip the molecule over and have a direction in one way and a direction the other way, like two lanes on a road. There's a great picture of this. Slide 25. Kind of shows you where the phosphate is, and those carbons in the ring are numbered. You'll see like an O. I think it's 1, 2, 3, 4. Yeah, that's right. One, two, three, four. And then there's another fifth one up here. Okay. And so we'll number these 1 or 3. And it's talking about these carbons in these rings that it's connecting to. So you can imagine, all right? I would just take a few minutes over the next couple of days and just look at these diagrams after you've read this simple paragraph here. One end has a phosphate, kind of get an idea of what's going on. Okay, you don't have to be an expert in it. So DNA itself, what's its main function? What does it do for us? It stores information, right? Info on how to make proteins. RNA has information. I want to say carries. As in transcription, like RNA will make a copy, come out of the nucleus, be transcribed into a protein. RNA has a couple other functions though. It can be an enzyme, an inhibitor, for example, has a lot of roles in the cell, not just carrying things outside for protein transcription. There's many different forms of RNA. You've got messenger RNA, transfer RNA, rRNA, ribosomal RNA. The protein building machines themselves that read the code are made of RNA. And if you remember when we were looking at the BAT paper way back last semester, that SSU single unit that they were looking at, that was all part of the ribosomal RNA, one part of the ribosome they took apart. Still a lot of stuff to discover there. Another nucleic acid, and the last one we're going to talk about, it's called ATP, and that is adenosine triphosphate. Okay. This molecule you can see on page 46, and you can also see it on slide 30. Has that double ring nitrogen base, the 5-carbon sugar, and the 3 phosphates, triphosphates attached to it. I'm going to draw a crude representation again. Okay. Now, if you had to guess, here's our base, our other two bases. Which one of these phosphates carries the most energy, is the most unstable? Yeah, the one on the end is the one that's peeled off to produce energy. When that bond is broken, energy is released. When that happens, it's only got 2 phosphates. We call it adenosine, or ADP, adenosine diphosphate. ATP is the energy currency of the cell. Cells use energy released by breaking down food molecules to synthesize ATP. So everybody understands glucose is what fuels cells, but it's actually glucose being broken down into this that is what's used. And I've told this to you before. If a cell is a vending machine and you only have a $20 bill, that $20 bill would be like glucose. You can't really use it. But if you go break it into quarters, the quarters could be considered ATP, and that's readily used by everything, right? So it's energy from ATP hydrolysis is used to drive energetically unfavorable chemical reactions to power transport across membranes. Okay. There's 2 other important molecules, NAD and FAD. These are electron carriers. That's nucleotide-containing molecules. We'll talk about those later in chapter 7 and 8. But still all basic components to come together to make all these molecules that do all this crazy stuff for us, just atoms. Water, simple hydrolysis, dehydration reactions, stuff like that. What holds the 2 strands of DNA together? I didn't talk about that. What kind of bond? It's on slide 29. You see that G and C right there and the little red dots in between it? What kind of bond is that? Hydrogen bonds, yeah. So you see why a dehydration or hydrolysis reaction would work perfectly to open that up, right? Okay, and to build polymers, release polymers, stuff like that. Yeah, I mean, it might be beneficial to know that like we know we build polymers by which kind of reaction? We talked about it at the very beginning. by dehydration, right? So if I'm taking one of these and peeling it off, I'm wanting you all to try to connect that that is a hydrolysis reaction because it's breaking things down. It's taking one apart, right? And to do that, a water molecule had to be produced. If I were putting one back on there, it would be a dehydration reaction. So for building carbohydrates, it's one of the two, to build or to break down. ATP, to put that phosphate back on there, it's one of the two, hydrolysis or dehydration. Okay, I want you to be able to make that distinction. All right, I think I've talked about that. We got two more to go. The next one is proteins. Any questions about DNA in terms of just the molecule, DNA, RNA? No? All right, give me a second to erase this. I gotta make some better magnets. Proteins. We'll try EIN, I think. I was right that time. Look at all the jobs proteins can do. They've got 7 listed here. As catalyst. Can you read them off for me? Defense.
Transport, support, motion, regulation, and storage.
Okay, some examples. For defense, that would be immune system. Okay, certain proteins can be activated to take care of some bugs in our system. Transport, chaperone proteins is an example. Some of your thyroid hormones can't be out there floating around in your blood unless they have a chaperone protein attached to them. Otherwise, they will just dissolve into the water. They have to be kind of protected until they're ready to be used or they're hooked up with their other substrate. Support, that would be things like collagen, keratin, and others. The most abundant connective tissue in our body. Keratin is what's made of our nails and our hair. Collagen makes up skin, ligaments, tendons, bones. Motion is referring to muscles. Okay, those protein filaments that pull to give us motion. Regulation, small proteins called hormones. We'll put that there. And storage, things like calcium and iron. There's a great table on page 47 that kind of lays this out. Proteins are what, the name of the game. What do we make proteins of? Amino acids. And once you get a bunch of those linking together, we'll talk about them. You get a peptide, and then as that peptide grows, it might become the starting of a protein. There's a lot of steps in there we need to talk about. Amino acids are pretty cool. Proteins are made, most proteins we care about are made with 20 amino acids. can see them right here. So think about every protein that makes up every organism on Earth. Just from 20. Flies, humans, bananas, whatever. All that variety comes from just rearranging 20 of these amino acids together in a long chain. These amino acids, you can group them by hydrophilic, polar, hydrophobic, nonpolar, negative charge, positive charge. So when they start linking up in a long chain, they start having these polars, they will start to fold over on themselves based on those reactions or attractions they have between atoms. All right? You're not going to have to know, like, this group is polar, this group is unpolar. But when you get in the weeds in biology and you start doing real work that is applied, it's important. For us, we want to know this because it's really what drives the ultimate end product of a protein. And I want you guys to know a little bit about what's going on inside here, okay? So proteins could be big units, right? Usually when a protein is done folding, the inside of that protein is going to be hydrophobic. It's going to be afraid of water, okay? That's a general rule. And the outside will be hydrophilic usually. There might be two L's in that word. So most amino acids have the same basic structure, and then they have these things called R groups that are attached to them that give them their unique properties. Okay, that R group just stands for any kind of group that can be put on there, and you'll see it in molecular formulas. It just represents like a group of atoms that go on top of it. So each amino acid affects the shape and protein differently depending on the chemical nature of its side group. Side group. Hint, 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 side groups. For example, proteins or portions of a protein chain with numerous nonpolar amino acids tend to fold to the interior of the protein by hydrophobic exclusion. Remember, most of this stuff takes place in a water environment inside of us, inside of our cells. And so if there's water around everywhere, that protein is going to be pushed together to where the hydrophobics are inside and the hydrophilics are outside. That's one force helping things fold the way they should. Amino acids are linked together by peptide bonds. That shouldn't be too far of a stretch because we're making a peptide, right? Peptide is another name for a short protein sequence. What kind of reaction do you think puts those peptide bonds together? Looking at this, keep referring back to it. Which one builds a molecule, dehydration or hydrolysis?
Dehydration.
Right. So they undergo a dehydration reaction to form a covalent bond. A covalent bond that links two amino acids is called a peptide bond. You get a big one going, you got a polypeptide. And actually we found this out, this is kind of neat. This guy named Sanger in the 50s had a clue that this was happening. There were certain amino acids in a protein, and he painstakingly removed one each from the insulin protein and figured out its sequence. Was able to start making it. This is back when we didn't have insulin ready to go. So that was a big discovery for people. That was like one of the first ones. Although many of these acids occur in nature, only 20 commonly occur in proteins. Of these 20, okay, 8 are essential. What do we mean by essential? Have to have what? Have to have them in our diet.
Yeah.
We can't make those. We have to eat them. We have to ingest them. So they're essential in our diet. Okay? Essential amino acids, 8 of them. All right, so let's talk more about how the protein folds. I highlighted something here. It says, essentially all the internal amino acids are nonpolar ones. Amino acids such as leucine, valine, phenylalanine. Water's tendency to hydrophobically exclude nonpolar molecules literally shoves the nonpolar portions of the amino acid into the protein's interior. All right, and so a protein is usually built or can be built in 4 main steps that we need to kind of categorize or characterize. Primary structure. This is the first step in making a protein. What defines a primary structure? The book tells us that the primary structure of a protein is its amino acid sequence. It said something about hydrogen bonds on here. Consider protein hemoglobin. Yeah, it's just the chain before it's done any folding. We'll call it that, okay? We'll just say it's a chain of amino acids. What characterizes a secondary structure? So there's no folding yet in our minds. No folding yet, just a chain. Had some folding here, okay? Amino acid side groups are not the only portions of proteins that form hydrogen bonds. The polypeptide backbone consists of repeating units of amino acids with amino and carboxyl group that can participate in hydrogen bonds. There they are again, hydrogen bonds. They can be with water or with other amino acids in the backbone. If too many hydrogen bonds formed with water, the protein would tend to behave like a random coil. It's talking about side groups. When they do start to fold, though, they got 2 structures that can fold. I think it's safe to put hydrogen bonds here. We got some. But when they do start to fold, they take on 2 of these structures that we call alpha helices or beta sheets. Helices is just like you think, helices, like DNA. And the proteins that interact with DNA are usually helical shape. The sheets are pleated sheets. So if I were to take a piece of paper and fold it like an accordion this way and I pulled it out, that sheet, that would be like a pleated sheet. It's a beta sheet. And they can go together all kinds of different ways. Those are 2 important things to remember for step 2 or secondary structure. Tertiary structure. What defines that? The final folded shape of a globular protein is called its tertiary structure. It has regions that have secondary structure and determines how these further arranged in space produce overall. Okay. I'm going to put final globular shape. Okay, just track with me here if you can. A protein is initially driven into its tertiary structure by hydrophobic exclusions from water. We understand that part, right? Okay. Ionic bonds between oppositely charged R groups bring regions into close proximity, and disulfide bonds lock particular regions together. The final folding of a protein is determined by its primary structure and the chemical nature of its side groups. You can look right here. Hopefully it's in here. Slide 39. C, item C shows that ionic bond between two there. All right. When two or more polypeptide chains associate to form a functional protein, the individual chains are referred to as subunits. And then the arrangements of those subunits together is termed, guess what? The last stage, quaternary. I'm going to write subunit here. A great example of this is hemoglobin. It's made of 4 subunit proteins together to be a functional protein that carries or binds oxygen and carbon dioxide with us. That's this quaternary structure. I'm going to skip over motifs and domains for now. We'll start back on chaperone proteins, but one way to remember this is like increasing complexity. So if you want to say this is letters, this would be like words, this would be sentences, and this would be paragraphs, basically, in terms of complexity and information that's in there. Motifs and domains are sort of like a common way these things come together in a class of proteins versus another class. But it's all right. I'll leave that at that. So chaperone proteins are another class that sometimes help bind when these things are folding together. They might have to come along and push one group next to the other because of water and that hydrophobic hydrophilic reaction. I talked about chaperone proteins here, transporting things, carrying hormones through our blood, for example. There's a picture of this thing. I studied this in my master's program. I thought that was cool. It's like a barrel. Protein will go down in there. It will be capped. It will do its thing, and then it will uncap, and that protein comes out. It's ready to go because that chaperone proteins work. When a protein is unfolded because of increased temperature or pH problems or something like that, we call it a denatured or denatured. Okay, proteins can become denatured or unfolded, and typically we would think, or we do count on them being able to fold back once those conditions are fixed in a way. So they should go back to their original shape. I think that covers mostly what I want to say with proteins. All right. So we're going to pause here in a minute. I'm going to show you, we're going to do the little amino acid worksheet. But one wrong base can equal one wrong amino acid, which can equal a malfunctioning complete protein. Case in point is, what's the one where the blood is shaped like a sickle cell disease? Okay, the hemoglobin molecule is off by one amino acid, and it causes the proteins kind of become sticky and makes the cell fold over on its shape and take on a sickle shape. That's from one base being wrong. Cause a lot of damage. Any questions about that? Okay, I'm going to pause there. Helps me match up the two videos if I want to merge them later. All right, lipids. Long chains of last molecule lipids So these are long hydrophobic chains of carbon and hydrogen bonds. And they're a lot longer than carbohydrates. Remember, carbohydrates are long chains of carbon too. Carbohydrates have different functional groups up here. Lipids are long chains of carbon. Usually, I think in books say like up to 40. Anywho, they're insoluble in water, meaning they're hydrophobic, and that's why you see, insoluble. What does insoluble mean?
It's not gonna, it's not gonna
Break apart, dissolve,
Thank you.
right? Right, you put oil into water, it doesn't mix, right? It separates. There's something in this paragraph. A steady place of water, many lipid molecules spontaneously cluster together and expose a polar hydrophilic groups that they have the surrounding water while conforming nonpolar parts of the muscle or molecules together within the cluster. All right. Many lipids are built from simple skeleton made up of two kinds of molecules, fatty acids and glycerol. Fatty acids are long hydrocarbon chains with a carboxyl group at one end. And glycerol is a 3-carbon polyalcohol. It has 3 OH groups. All right, so this is glycerol, and this is what we call a fatty acid. This is usually the hydrophilic end, the glycerol, and this will be hydrophobic. Okay, just back end here. And that's what makes them clump together in a water solution also. When they clump together, you usually get 3 of those fatty acids on a glycerol head. That's what we call a triglyceride. Has anybody ever heard of that before? You ever heard of triglycerides? When you're my age, you'll start worrying about that. It's cholesterol in your blood. It says right here, it says the most common even-numbered chains of 14 to 20 carbons. Okay, these CH bonds of fat serve as a form of long-term energy storage. Okay, so if you get 3 of those chains together, it's a triglyceride. See a picture right here. Notice these, if one of these hydrogens are missing, we would call that an unsaturated fat, okay? When they're all together, when every carbon has its hydrogen bond, it's called saturated. All right? So saturated fat. An unsaturated fat might be missing a hydrogen. Causing a carbon-to-carbon double bond. Okay, right now there's no double bonds in this straight, solid chain. But when you take one out, you get that double bond, it allows it to kind of twirl around this bond here. And that makes the chain bent. Okay? And if it's bent, it's unsaturated. This is why saturated fats are solid at room temperature, like butter, margarine. All that stuff has been, most of it has been hydrogenated, so it's stable and solid and doesn't spoil. But unsaturated fats are liquid at room temperature.
So like lard, that would be a saturated fat?
Yep, exactly. Let me read this to you here. When I was reading this over the weekend, I thought this was kind of funny. So it says, for many years, the consumption of saturated animal fats was linked to increased risk of cardiovascular disease. Because of this, the food industry started producing solid products, such as margarine, by adding hydrogen to unsaturated fats to make them partially saturated. This process, called partial hydrogenation. If you ever look on the back of a tub of cream cheese or butter or something, it's going to say hydrogenated or something like that. They have made it like this, so it's not because it was unhealthy. It's because it was shelf stable, and they could ship it out to stores and it not go bad in a week, and like they would have stock to sell still. It's profit. It's a profit game, is what it is. That's why it was built. Unfortunately, the process also converts cis double bonds to trans double bonds. Don't worry about the cis and trans for now. Creating so-called trans fats. Because they are less kinked, they are solid at room temperature, but also have been linked to elevated low-density lipoprotein, the bad cholesterol, LDL, and lower levels of the HDL, the good cholesterol. So the risk for cardiovascular disease from trans fat appears to be worse than just regular saturated fat. And I think just in the news recently, they flipped the food pyramid and said, eat more butter and meat, right? Eat saturated fats. That's what they're saying. So they tried to make something better, but it was really worse, and I don't think they were really trying to make it better. But placed in water, triglycerides spontaneously associate together, forming fat globules that can be very large relative to the size of regular molecules. This is the oil beating up in water, okay? Organisms contain many other types of lipids besides fats. Terpenes are long chains of lipids. They're biologically important in pigments such as chlorophyll and visual pigments in the retina. Terpenes. I think, if I'm not mistaken, something about marijuana, THC, that's a terpenoid. It's like a fat of the plant. That thing that gives us that high. Steroids are another class of lipids, okay? So terpenes, I'll put chlorophyll and pigments. What did I just say? Steroids. Steroids are another class of lipid, and they're composed of carbon rings, and most animal cells contain steroid cholesterol. That's like a structural component of the cell membrane. Kind of gives it a little bit of rigidity. Other steroids, such as testosterone, estrogen, function as hormones. Prostaglandins are a group of about 20 lipids that are modified by fatty acids. Prostaglandins act as local chemical messengers in many vertebrate tissues, like when you have an inflammatory response from a splinter. It's prostaglandins. Fats are excellent energy storage molecules. Okay, you remember how I said carbs had 4 kilocalories per gram, right? And lipids have 9 kilocalories per gram. Why is that? There's a lot more carbon-hydrogen bonds, all right? They're more dense. So nature chose lipids, this dense form of energy storage, over the carb energy storage, because you can get more power for your punch compacted into a small size. This molecule is basically without any water in it. There's no, it's what we'd say is anhydrous. But carbs, they have some water molecules playing around in them. And that water carries weight, right? So I think I can remember this math. If I'm a 200-pound man, and I typically would carry fat about 25 pounds, that's all my energy storage. That's about 100,000 calories I have stored. Okay, if I wanted to have that much calories stored in carbs, it would be almost twice as much. So it would be about 50 pounds. And then if you were to put the water factor in there, that would put my weight, if I was a 200-pound man, up to about 400 pounds if my energy storage was in carbs versus lipids. They're compact, they're energy storage in them, okay? That's what I want you to remember about fats versus carbs. So 4 and 9 kilocalories. Lastly, we got these things called phospholipids. Everybody good up here? Okay, phospholipids, instead of the trans or the triglycerides, they have 2 chains, 2 carbon-hydrogen chains on a phospholipid head. Okay, this head is hydrophilic, and these tails are hydrophobic. Why do we need a molecule that's both hydrophilic and hydrophobic? What happens to fat molecules or lipids in water? They clump together, right? Okay. Because of this configuration, when they start clumping together, they line up like this. And if you were to do that in a complete circle, you would have what we call a me cell, which is like a tiny proto-cell membrane. You've got this interior where all these hydrophobic tails are pointing to, and this exterior now because of the hydrophilic heads. Now we've got a barrier where a cell can happen versus just a soup of molecules, right? Cells are particular because they have a bilipid layer. Their layer is not just one, they'll have 2. So all the way around. And that's called a bilipid membrane. Okay? Has 2 layers. Page 58, figure 3.31. Form all biological membranes. Can't be said better than that. Any questions? So if we start looking at this, how did the first cell arrive? Like the origin of life questions I gave you. How did this happen? Well, we had to have carbon, we had to have hydrogen, we had to have water, right? Phosphorus, nitrogen, all the things on Earth. And this could spontaneously happen in water if you got enough lipid molecules together. It can make a me cell. But how does that turn into a bilipid layer? And how does that layer get certain proteins that allow things in and out and these pumps and stuff? Nobody knows for sure. You can see some people think the membranes form first. And that solves the problem of a cell having to have an inside versus an outside. It has to be compartmentalized. But you don't get any of the metabolism machinery inside working, or no genetics either. Did RNA come first? Well, RNA is unstable. It doesn't last long in a solution, so it has to be inside of one of these. But some people think RNA came first rather than the cell. Some people think the metabolism, like the actual making of ATP, came first and cells were around that. That would be things like metabolism first. The clay mineral surface probably too. Everybody's probably heard of the primordial soup before, right? The Urey experiment where they took a bunch of amino acids or nucleotides, put them in a solution and boiled it and zapped it with electricity, trying to simulate like early Earth, lightning hitting water, causing these things to spontaneously form. That's pretty much dead in the water now, because if you look at systems and you look at this electron transport chain and how cells are moving hydrogen around to do work to make ATP, I forget where I was going with that, but this book right here explains that it's metabolism first, and I kind of subscribe to that. This guy's a biochemist, he's a famous one. That's why I started reading over Christmas break. But he thinks metabolism started in sea vents down below the bottom of the ocean, not the hot ones, but the ones where cold water's coming up. And they form like these thin eggshell kind of caves on the bottom of the floor with holes in them. And water is coming up from the mantle of the earth that has certain chemicals in it, and it's meeting this acidic ocean that's early earth with its components as well. And it's kind of flowing across this shell of crust on the bottom of the ocean that has metals and minerals in it that can catalyze these things. And when that happens, you might get one of these to form around one, and then it comes off and becomes a cell. Thousands and thousands of steps in between there that have to be answered, but I'll digress on that. We'll say intelligent design is the only one that does answer all of these, even though it's not testable or scientific. It does provide a solution for all of that in a certain way. Any thoughts, any questions? Okay, so we went over a few basic ideas about our molecules of life: lipids, nucleic acids, proteins, carbohydrates, and some key things about those. Okay, I think I'll end it there, and then we'll discuss how we want to move on from